So, in this video, we'll be covering lactic acid fermentation, a type of anaerobic respiration. But in order to understand anaerobic respiration, we must first understand aerobic respiration. So, under glycolysis here, we can see that we have glucose, a 6-carbon molecule, being converted into 2 pyruvate molecules, each with 3 carbons. And when we do this, we see that we have 2 ADP plus 2 inorganic phosphates here, being used to create 2 ATP. And this is how our body creates energy. But we can also see that we have down here, we've got NAD plus being reduced into NADH. We actually have 2 of each of these. And these become really important later in anaerobic respiration. So if we move along here, you can see that our 2-pyruvate converted into 2-acetyl coenzyme A, which are then pumped right into the Krebs cycle. And in this cycle, we can see that we have 2 more ADP being converted into 2 more ATP. But we also see that we have a lot of NAD plus and NADH being converted. So we actually have 6 NAD plus being converted into 6 NADH, 2 FAD being converted into 2 FADH2. And these molecules here are our electron transport carriers. So we can see that our NADH and our FADH2 are pumped right down into this electron transport chain here. And the way this works is we're actually oxidizing NADH and FADH2 into NAD plus and FAD. And when we do this, we see that we trigger hydrogen pumps. And what these do is they send hydrogen across the mitochondria, out of the mitochondria, where they're then eventually pumped back in through an enzyme known as ATP synthase, which then converts a total of 32 ADP and inorganic phosphate into 32 ATP. And this electron transport chain here, this is where we actually create most of our ATP in our cell in the aerobic respiration. But if we look at this, we see that down here we have oxygen. And we see that the oxygen is actually accepting the electrons as they come down through this electron transport chain. And what it's doing is it's collecting these electrons and it's forming water. And that's why we know oxygen to be our final electron acceptor here. But what happens if we don't have oxygen, or if we're an organism that hasn't evolved to use oxygen yet? Well, in this condition, we have 2 options. We can either produce ethanol, or we can make lactic acid. And I'll show you why this works in a minute. So, when we convert our 2-pyruvate here, this is a pyruvate molecule, into lactate or lactic acid, just depends on how you have the hydrogens laid out. So we use an enzyme known as lactic acid dehydrogenase to perform this reaction. And what we can see here is that we're actually we're reducing pyruvate to produce lactate. And in this process, we are then oxidizing our NADH to produce NAD+. And this is really important because if we look back here into the electron transport chain, we can see that if we don't have any oxygen to accept the electrons, this is going to stop. It's not going to create any more ATP. We're not going to have any ATP produced in here. We're also not going to have our NADH oxidized into NAD+. So eventually, we're actually going to run out of our NAD+ and our FAD, and that will actually stop all of these reactions over here, including glycolysis, including Krebs cycle. All of them will be stopped. See, because they both need NAD+ to carry out the reaction. So when we convert our 2-pyruvate here into lactic acid, what we're doing is we're really just using an alternate method to produce NAD+, so we can continue to make these 2 ATP up here. So our body can continue to function when we don't have enough oxygen. Say if we're really exerting our muscles, they can run out of oxygen. We can't pump oxygen to them fast enough, they have to undergo lactic acid fermentation. Now, we actually have some uh, uh, actual uses for these in like the food industry. So, using bacteria known as lactobacillus here, these are what we use to produce yogurt, and variations of these are used to produce sauerkraut and I think kimchi, the Korean food. And so for the production of yogurt, we actually have our milk, which contains lactose, a disaccharide sugar containing galactose and glucose. And so what lactobacillus does, so what lactobacillus does here is it will split up the lactose, and it'll use the glucose in glycolysis to produce pyruvate. Pyruvate is then converted into lactic acid, right? And now this lactic acid here, it will reduce the pH of the milk to the point where the proteins in it start to denature, and depending on how low you bring the pH, how the temperature of the milk, and the amount of proteins in the milk, so it's dependent on many factors, but depending on those factors you'll get a very unique type of yogurt, right? So unique depending on each batch. You may be wondering why we still use the metabolic process that is given up here, so glucose to pyruvate to lactic acid, done by just done through anaerobic respiration. It's very complicated, a lot of factors involved in it, and you may be thinking there's a better way, and there are other alternatives in for producing lactic acid, but they have their pros and cons. So the metabolic process, the main reason why it is still used is because it's much cheaper than the other options, and it's been explored more. This is how they started producing yogurt, so it's a tradition, and I don't know, it just seems a lot safer to be using an actual metabolic process to produce our food than using harsh chemicals, including catalysts like lead ions, which we'll see in the next reaction. So it's also safer for human consumption. But the metabolic process doesn't come without its drawbacks, and these are that it is you get lower yields, right? So generally in the range of a little bit less than 30%, right? So about 30% yieldish, somewhere around that, and the efficiency is very low, right? And you also have to run the reaction at higher temperatures, right? And with higher temperatures, you have to pay to heat up your reactants to that temperature, and that costs some money, and it wastes energy. So if we go down here and we take a look at the synthetic process that we use, you can see that in this example, we're starting off with cellulose over here. Starting off with cellulose, and we're converting that, so we're hydrolyzing that into glucose. So we're splitting the cellulose down to produce glucose molecules. And the glucose is then catalyzed by the lead 2 up here, as I was talking about earlier. And what it does is actually breaks this down into lactic acid, as well as some other byproducts, but those aren't relevant here. So that's the synthetic method that we're going to be looking at. And benefits of this process, of the synthetic process, include that the efficiency. So it's very efficient compared to the metabolic process. So the study reported that they had greater than 67% yield, which is a lot higher than the 30% yield that we are expecting from the metabolic process. And this efficiency is expected to increase as more research goes into this. So, you can perform this reaction under very controlled environments. You can control all of the factors going into it. So you have a high level of control of the actual reaction. Now, downsides of the synthetic process of producing lactic acid are that it's very expensive in comparison. So this is much more expensive than just using bacteria. And also, there's worry of the byproducts that it releases, right? Because the glucose doesn't just produce lactic acid, you also produce some other complicated molecules. So byproducts, you might not want that in your food, so you'll have to find a way to remove those.